So just to add to who I am, so my lab is focused on various types of imaging technology developments, specifically in the last sort of decade, image-guided interventional systems, so biopsy and therapy guidance systems using medical imaging. So this particular project is a new project in adaptive oncology, and that is integrating a biopsy guidance mechanism into a PEM, a positron emission tomography system. So it's a positron emission mammography that's clearly focused on breast biopsy. So the rationale for this kind of development is that current breast biopsy strategies are not yet quite optimal, especially for ultrasound occult lesions. Ultrasound is used to guide needles into the breast, in fact, for all biopsy and some of interventional procedures. But some breast lesions are not visible under ultrasound, and therefore these women have to go to MR and then biopsy procedures. Some centers are developing various other mechanisms for biopsy, but basically these women have a more complicated path. There's a need for improved biopsy mechanism or approach for these cases, and especially for heterogeneous tumors where one wants to sample a tumor in multiple sites, and therefore one needs accurate guidance, as well as, of course, detection, a sensitive and specific method to detect the tumor, and then accurate guidance of the needle to a specific target. So positron emission mammography has the potential to provide this kind of capability. PET, positron emission tomography, is a whole body imaging system that many of you, all of you may know about, that's used to image tumors in the body, wherever they are. But those kind of systems can be used to image the breast, but they're not optimal because they're optimized for whole body imaging. Some companies, a few companies and universities have developed positron emission mammography system. So these are specialized PET systems for imaging the breast. And therefore, we are proposing to improve biopsy guidance by adding a biopsy mechanism to these PEM systems. So the opportunity is to combine the strength of our OACR imaging program that we have across Ontario and leverage the development of a PEM system at Lakehead University with our development of image-guided interventional system to provide a PEM-based accurate biopsy guidance mechanism. So our thinking is the workflow will be as described. So PEM has the opportunity to provide diagnosis, of course, but as well as treatment planning and monitoring response. But in our case, we are focusing more on the biopsy capabilities. So suspicious lesions will be identified they could be identified with ultrasound, or in fact, if the ultrasound is not able to identify a lesion, then women will have to undergo a PEM imaging procedure, positron emission mammography, and then biopsy immediately as part of the system, guide a needle to a target to sample, to provide a diagnosis, and of course, sample the tumor. So this is an example, as all of you know, the conventional mammography is on the left side, and a PEM image, this courtesy from a company that's manufacturing a system like that, by company's Naviscan. So we can see that lesions can be identified quite well with these kind of PEM systems. In this case, as you'll see later, it's the image on the bottom right is a PEM system that's been developed by a company out of Thunder Bay called Radialis. And it's the principal and developer is Ala Resnick, who's a collaborator on this project. So we are proposing to deal with the unmet need to improve spatial sampling of breast tumors, which requires improved detection and guidance of biopsy needles to a target. And the underlying hypothesis is that the PEM approach will provide improved detection small tumors and provide improved guidance of a needle to a target and to allow sampling of a tumor. So this is a multi-institutional collaboration with a company as I described. So the aim of the project that we've been working on for the past few months, I guess it's now 9 months, is to develop a clinic-ready PEM-based biopsy system. So to add a biopsy mechanism to a PEM system that's already been developed by Radialis, and this will allow improved tumor detection. So this kind of technology development has got it's quite a big, it's quite extensive. It requires development of hardware for biopsy of a biopsy subsystem that's very accurate. Develop a guidance software to allow guidance of a needle to a target with high accuracy. Of course, test the whole system before we get to patients. We have to test the system with phantoms before we can get Health Canada approval, and then integrate, of course, integrate the hardware and software subsystems into the PEM system and then test them clinically. So our proposed deliverable is to have a clinic-ready PEM system for improved breast cancer detection by a PEM, and of course improve biopsy or accurate sampling of heterogeneous breast tumors using our mechanism. So just to review a little bit about the PEM system, because of course it's central to this project. It's, as it said, developed by Radialis, a company out of Thunder Bay. So it is based on some new technologies. The detectors are high-yield cerium-doped lutetium yttrium orthosilicate scintillators. You see the picture on the right-hand side. It's an array of crystals that have a very high detection efficiency for the radiation, the annihilation radiation in PEM PEM systems, and then coupled through a light guide to a detector array. So again, there's on the left side is a picture of a detector array. It's a tiled array to provide large area, a large area. The area, as you see, is 17 by 23 centimeters. So similar to a mammogram system, a mammography plate system. So this kind of approach has been already tested by, and you can see the pictures on the, here the resolution is, and sensitivity is quite good. The PEM system is shown on the right-hand side where you can see the plates are positioned over the breast. They are not they're not, in fact, the breast is not squeezed like it is with mammography. It doesn't need to be squeezed. In fact, they're separated by about 8 millimeters or larger. So basically, they have stabilized the tissue, and then the image is collected. That's an important aspect because we need to be able to get the biopsy mechanism between the plates to access the breast. So the current PEM system now commercially available produce the image on the left, and the Radialis system is a much higher sensitivity, and you can see the result on the second picture. So this is the system we're adding our biopsy guidance to. So we have been developing various kinds of biopsy guidance mechanism over the past many years, decade or so. We have developed and licensed to many companies various technologies, and some of them are used now worldwide. We have developed a prostate biopsy system that's used to guide needles into the prostate for biopsy and therapy. We have developed a liver ablation system with mechanism to guide needles into liver, and then also for guiding gynecological brachytherapy needles into gynecological tumors. So we have a fair amount of experience with mechanism, hardware and software mechanisms to guide needles into tissues using ultrasound guidance, which is a less expensive approach to imaging. So we've leveraged all this technology. We have developed and filed patents on counterbalanced mechanism, which makes any device weightless. It has inertia, but it is weightless, and again, we filed patents on all these. And then using fusion software, we can fuse the PEM image to ultrasound image. So we have a fair number of various fusion software modules and 3D visualization and 2D and 3D visualization modules. So basically we're assembling a complete system based on some of these technologies and that we've developed in the past. So the mechanism we have developed, so on the left side is the design of the system, is the core system. You can see the counterbalanced support arm. So this is the patent file that makes the whole system weightless. And better not touch it. So the end effector is the ultrasound transducer, ultrasound probe, and the biopsy device. So this is a unique design. It has a remote center motion, which means that the center line of the ultrasound transducer and the needle, the biopsy, the center of the core coincide, which means if we place the mechanism such that the remote center motion is centered on the lesion, then we can guide a needle to a target. On the right-hand side is the system we have built. So this is now, as you can see, the biopsy device, the ultrasound transducer, and the complete device. So we have not yet built a counterbalance support arm, but we built the biopsy guidance subsystem, and we are testing it now. So the way it will be used, as you can see on the left side, is the PEM system, and the plates are in place over the breast, and then the guidance mechanism is inserted between the plates to guide the needle to a target, which is visualized with the PEM system, and then registered with the ultrasound image, and then the needle is guided to the target. So there is a closer view of on the right-hand side of the simulated approach. We can see the ultrasound transducer and biopsy guide, and on the left side is an experimental simulation of the system, where we have targets in the agar phantom. So it's a tissue equivalent agar phantom that's ultrasound compatible, and we've placed a number of targets in it, and then we simulate the procedure just to test the accuracy of our device. And there's an example of some of the experiments we've just done. So this is with the 6mm targets. You can see on the left side multiple targets, and then we fire the needle into the phantom, and you can see the track of the needle going through approximately the center of the target. And so of course we're repeating this for many trials, many sizes, different locations, different depths. And on the right side is another example with 3 needle passes. So these are early results. We show the accuracy of our biopsy guidance system, where we plotted the error in millimeters on the vertical axis and the depth of the lesion ranging from 4 centimeters to about 6.5 centimeters. So you can see the error in guiding a needle to the target is about 0.2 to about 0.8 millimeters. So that's in fact quite good. It's of course these are under optimal conditions. Of course, there's no movement, no breathing. It's a static phantom under idealized conditions. So we certainly, we don't think we can do better than that, but of course we have to build a complete system and to see how all the errors and the mechanisms propagate to give us the final error. So, so this gives you a quick overview of the project and where we are. So we've completed the design of the complete system. We filed patents on the counterbalance arm. We have developed a biopsy simulation experiment already, and we have now preliminary results are quite good. But there's a fair amount of more work to be done over the next 12 months. We have to complete all the biopsy experiments to see the accuracy and under various conditions. We have to build a counterbalance arm and test that subsystem. We have to develop all the fusion and guidance software. Some of it has already been done, but we have to integrate it all into the complete system, and then use the complete system to test the accuracy of a guidance system, and before we even get to the PEM system. And then once we are satisfied that our design is accurate, then we can integrate it into the PEM system for the LS. So this is a quick overview and where we are, and hopefully sometime later I can update everybody on some of the more recent the work with a complete system. So thanks for your attention, and happy to answer any questions.